Today, let's dig into what makes proteins take on the shapes they do, because structure leads to function, and protein functions make up many or most of your cellular physiology. Amino acids have three parts: an amino group, a carboxyl group, and an R group. These are all attached to a center carbon. Peptide bonds form between amino and carboxyl groups, leaving the R groups to interact with each other and their surroundings. From weak to strong, the interactions holding proteins in a 3D shape are hydrophobic interactions, van der Waals forces, H-bonds or hydrogen bonds, ionic bonds, and disulfide bridges, which form between the cysteines in the R groups of cysteine amino acids. Of those, only H-bonds and covalent bonds are really stable in water. The rest of the interactions happen inside the protein. And amino acids exist in many forms: hydrophilic, hydrophobic, ionic, both positive and negative, polar and nonpolar. Check the end of the R group of an amino acid to help you determine the characteristics. Look for functional groups you recognize, or rich hydrocarbons generally indicate nonpolar molecules. So the sequence of the amino acids determines how the protein will fold because it determines which R groups are present and where. That sequence is primary structure. Many of our proteins have sequences that will form one or two common and easily recognizable structures: alpha helices and beta pleated sheets. Proteins can have one, both, or none of these structures. We call them secondary protein structures. They're like a building block for the larger 3D structure. That globular 3D shape built from a single strand of amino acids is the tertiary structure. Look at these ribbon diagrams. You can see the secondary structures as ribbons, and the wiry parts of the protein are the other parts of the protein strand. Quaternary structures sometimes form. They are built from many tertiary structures coming together for one purpose. I think of hemoglobin or ATP synthase as examples, but there are many more. So all proteins must have a primary and a tertiary structure. Secondary and quaternary structures often happen, but it's not required to make a three-dimensional globular protein. The hydrogen bonds and other forces that hold their shape intact are delicate. Substituting one polar R group for another one, or one hydrophobic for another one, may not change the shape or function of that protein, but changing a polar R group for a nonpolar R group means that nonpolar amino acid might try to dive into the interior of the protein, causing a potentially big change to overall shape and function. In a similar way, binding a molecule to a protein can also disrupt the bonds holding the tertiary shape in place. As the protein changes shape, it might be activated or inhibited. All those changes regulate a whole bunch of other bodily functions, so I hope you'll join me in showing some gratitude for the awesomeness that is protein structure.